Okay, let's take a look at Chapter 3, Objective 2B. So we talked about Objective 2A, which had to do with basically the phospholipid bilayer of a cell and the components of it. But now let's take a look at how materials move through that phospholipid bilayer. So this is regulation of materials through the cell membrane. Now, the cell membrane can regulate two different methods of moving materials into and out of the cell. Substances can pass through by using what's called passive transport, and that's just another way of saying you don't need any energy. I'm going to tell you folks to put this in your long-term memory because we're going to come back and talk about passive transport and active transport when we get to Chapter 12, the nervous system. So this is long-term memory stuff, so keep this around for a while. Passive transport just means no energy input. Now, when we use the word energy, keep in mind the energy we are referring to here is ATP. So when we say that it does not require energy, what we're saying is it does not require ATP. Now, active transport, on the other hand, does require energy, and that energy is in the form of ATP, adenosine triphosphate, and that is needed to move the materials from one side of the membrane to the other. Now, if you remember from organelle discussions, ATP is made in the mitochondria, so right here we have a mitochondria, which is one of the organelles located within the cell. And this is where ATP gets made through a process known as cellular respiration. So now let's just kind of take a look at passive transport. We'll save active transport for the next video. But let's take a look at passive transport. There's three ways materials can move without using ATP from one side of the cell to the other, and they are simple diffusion, facilitated diffusion, and osmosis. Before we get into these, let's just kind of review what diffusion is. This video link is within your PowerPoint, so I encourage you guys to take a look at that so you can visually see if you need to. But without the video, let's go ahead and take a look and see what diffusion looks like. Now, diffusion is, by definition, I have it here in red, movement of substances from an area of high concentration to an area of low concentration. So you can see what this person is doing. They're putting some dye molecules into some water, and I know your instinct would be to stir it up, shake it up, something like that, to get those dye particles to equally distribute throughout the entire water within that beaker. But if you just let it alone and leave it sit, it will eventually completely move throughout the water and equally distribute themselves. When it reaches that point where it is equally distributed throughout the medium, in this case water, it's reached what's called equilibrium. Now, these are particles moving through a water medium. You could have particles moving through an air medium, such as if you were to take a air freshener and spray it in one corner of your room, eventually you would be able to smell it in the other corner of the room. It would be, for instance, if someone was cooking bacon in the kitchen and you were asleep in your bedroom and you could smell the bacon cooking. That smell was moving through the air to get to your bedroom. So particles, it doesn't have to be through water is what I'm saying. It could be through air as an example. So it's the movement of particles from an area of high concentration, like where this dye is accumulated, that's high concentration, to an area of low concentration. Another way of saying that, there's a couple of ways of saying this, which makes it a little confusing sometimes. It could be moving down its concentration gradient or moving along its concentration gradient. So all of those mean the same thing. You're going from high to low. Again, another video, I think it's the exact same one on the previous slide, just showing you visually what's happening if you needed to see that. So now let's take a look at the three methods of passive transport: simple diffusion, facilitated diffusion, and osmosis. So with simple diffusion, and I've put right here, yes, this is passive transport, meaning no energy is required. What we have here is the phospholipid bilayer. You can see the extracellular fluid. This is on the outside of the cell. You can see this is the cytoplasm. This side is going to be on the inside of the cell. So initially, we have all of these particles in high concentration on the outside of the cell. Now, these particles, some of them, depending on what kind of particle it is, can go straight through the cell membrane to an area of low concentration inside the cell, the cytoplasm, until equilibrium is reached. and that is when you have the same amount of particles on both sides of the cell. So with simple diffusion, particles can move directly through the phospholipid bilayer from an area of high concentration to an area of low concentration. You need to know the examples of materials that have the ability to move directly through the membrane. Not all substances can. So things such as gases, oxygen and carbon dioxide gas can go straight through the membrane. Your steroid hormones, estrogen, for example, testosterone, these can move straight through the membrane. Now, while they're larger particles, why can they go straight through the membrane? For that matter, why can fatty acids go straight through the membrane? Well, what's similar with all three of these substances right here? They all love fat. So they are what you would call as hydrophobic particles. They don't like water. So keep in mind where that hydrophobic zone is of your phospholipid bilayer. It's those fatty acid tails. So estrogen and testosterone and fatty acids have no problem moving through because they're not afraid, so to speak, of that fatty acid tail area that makes up that phospholipid bilayer. So they can easily move through. So you do need to know your particles there. This is just a video showing you those particles moving directly through the membrane in case you need to see that in motion. Facilitated diffusion. This is another type of passive transport, the way materials move from one side to the other side. So how do we deal with particles, for instance, that love water, that are hydrophilic? How do we deal with those? Because they obviously do not like this fatty acid phospholipid area right there where the fatty acid tails are. So water-loving particles aren't going to love going through that yellow section right there. So how do we get them through from one side to the other side? Well, there are these big proteins represented in various shades of purple here, in which particles can pass through. So they can use those as kind of like a tunnel to get from one side of the cell to the other side of the cell. Examples are going to be those hydrophilic molecules, those ones that love water, such as glucose, sugar, and amino acids, the building blocks of proteins and the building blocks of carbohydrates. Those are going to need a helper molecule to get them from one side of the cell to the other. And I've got a little video here to show you guys. Again, take a look at the video. This big purple protein molecule can move materials from outside to in or from inside to out, so it doesn't have to go one direction. And I'll let you take a look at that video on your own. But that is hyperlinked within in the PowerPoint. Okay, before we talk about the next one, which is osmosis, there's a few things we need to remember first, and that is, what is a solution? We learned this in the previous chapter. So a solution is a mixture of a solute and a solvent. The solute would be something like salt or sugar that gets dissolved in a solvent. And for our purposes, the solvent will be water. Okay, that's the most universal solvent, especially when dealing with the human body. So when you take something like salt and dissolve it in water, you get a saltwater solution. So just remembering those terms. So what we're going to do now is, in your handout, in your packet from the bookstore, let's go ahead and take a look at that. And I'll tell you real quick what page I'm on. I'm going to click that ladybug so we can do a little drawing now. Okay. So I'm on page around 53-ish or so is where I'm at. And we're going to take a look at tonicity. So on the very top part right here, we have solution A and we have solution B. The fact that there's 100 milliliters of water, your solvent, in each beaker is irrelevant. The big thing was, I was just trying to show you that the solvent's the same in both. Other than that, we're not going to worry about it. What we're focusing on are the solute particles. So these big, huge particles right here, these are representing the solute. And you can tell that in solution A, you have less solute than you have in solution B. Solution B has more solute. So because of that, instead of saying, well, solution A has less solute, solution B has more solute, we're going to use these terms right here hypotonic and hypertonic to describe the amount of solute present in those containers. So what you see is if you've got one substance that has less solute than another, you would say it's hypotonic. If you have one solution that has more solute than another, you would say that's hypertonic. So let's take a look at how this could be worded. Solution A is blank to solution B. Well, solution A has less solute, so we would say solution A is hypotonic to solution B. Now, you can only use these words, hypotonic and hypertonic, when you're comparing two things. So in other words, I couldn't look at one solution and say, oh, that's hypertonic. You don't know because you don't have anything to compare it to. It's kind of like saying, wow, that tree's really short. Compared to what? You have to compare it to something else, right? So solution B, you could flip that around and say solution B has more solute than solution A. So solution B is hypertonic. Now, if we were to take a look at the next picture here, we see we have two solutions, solution C and solution D. Again, the water's irrelevant, but that is your solute. And we just have the same amount of, sorry, that's a solvent, the water's a solvent. You can see that we have the same number of solute particles in each container. So we can't say that one has more than the other because they're the same. Anytime you have, iso means same, by the way, tonic means tonicity, which is referring to the amount of solute particles. So we could say solution C is isotonic to solution D, or we could say solution D is isotonic to solution C. So you need to understand these three terms when we're talking about tonicity. Okay, the next thing we're going to look at is page 54 on your handout here. We're going to look at what's happening up here in this first picture. Then we'll come down and take a look at what's happening in this smaller picture. So you have right here a container. So what we've done is we put two containers together. So you could call this solution A, and here's your semi-permeable membrane right here, and we will call what's on the other side solution B. You can see they have the same amount of solvent of water, okay? But what they have a different amount of is solute particles. So in this case, our solute particle is going to be a sugar molecule, and it's these big, huge spheres right here. These things right here, these are all sugar molecules. I know it's kind of hard to see in your handout because it's a black and white handout, so you can't really see the colors. So these are all your sugar molecules. Now all of these molecules here, these are water. These are representing your water molecules, okay? They're just little circles. So what you're seeing here is these sugar molecules, they're just too darn big to pass through and reach equilibrium. So your sugar cannot cross that semi-permeable membrane. It's just too darn big. So what's going to happen instead is we have osmosis that's going to occur, which is specifically the movement of water. So where diffusion is really talking about any substance moving from an area of high to low concentration, when we're using the word osmosis, we're specifically talking about water that is moving from a high water environment to a low water environment. And you're like, but wait a minute. There's the same amount of water on both sides. It appears that there is, and there is the same amount of water on both sides, but solution B's got something else going on here with the water molecules. But what you see here, see this arrow right here? This is representing the movement of water. So water is moving from solution A into solution B. So let's look at tonicity for just a second. Comparing just the tonicity, looking at the solute particles. the green stuff, the sucrose molecules. Solution A, how many sugar molecules do you see in solution A? Well, you don't see any. So this would be hypotonic to the other solution. If you look at solution B, or this side, you've got a lot of sugar molecules, sucrose molecules, in comparison to the other side. So this side is hypertonic. So because the sugar can't move, water does move. And water is going to move from a hypotonic environment always into a hypertonic environment. So let's take a look at why that is. So in the next picture here, again, pardon the fact that you don't see the colors, but you have the same kind of situation here. These right here are going to be your water molecules. They kind of look like Mickey Mouse, okay? They kind of look like a Mickey Mouse molecule. Remember, the formula for water is H2O. So you've got the one oxygen and the two white hydrogens that are attached to it, just to kind of show you what we're looking at here. So if you look, it has a low solute concentration. So this side is hypotonic. This side, in comparison, is hypertonic, because you can see that this side you have got these big, huge solute particles here. So these big, huge black spheres surrounded by water molecules, that is the solute, okay? And you can see that if you count the number of solute particles inside A, you don't have any. You have 0 solute particles. If you look at the number of solute particles inside B, you can see that you have 5 solute particles, okay? Now, let's take a look at the water molecules, which is the solvent. If you take a look and count how many water molecules there are on side A, you would see 24. If you were to count how many water molecules there are on side B, you would also count 24. So it would appear that these sugar molecules should move to the other side. They should go from side B to side A. That's what should happen. But they're bulky and they can't move. So water does move. If you look at how many of these water molecules have the ability to move and cross that semi-permeable barrier right there, every one of them can. They're all free to move from one side to the other side. Now, if you look at what happens with the side that has a lot of solute particles, you can see a lot of these water molecules are sticking to your solute particle. You've got these water molecules that are sticking to your solute particle. These are bound. These right here, these are bound water molecules. They cannot get away from that solute, unstick themselves, and move across that membrane. So the only thing you have available that can move are these water molecules right here. These are free water molecules, and they have the ability to move from one side of that membrane to the other. So you only have 4 free water molecules. So when you look at it this way, you have got a high free water concentration on side A and a low free water concentration on side B. So remember the definition of diffusion. We're going to make that the definition of osmosis, but it is the movement of a substance from high to low concentration. When we're talking about osmosis, it is the movement of a high free water environment to a low free water environment. So your net movement of water will go from a high free water environment, which is hypotonic because it doesn't have a lot of solute particles binding them up, to a low free water environment, which would be a hypertonic environment because the water molecules are being bound to the solute particle. So what does this have to do with what's going on inside of our bodies? Okay, let's take a look at, make sure I'm on the right page. I'm on the next page, which is osmosis in cells, isotonic. We're going to draw a picture here, and we're going to see how this applies, this concept of osmosis applies to what's happening inside of our bodies. So we have a blood vessel. Pardon my drawing, I cannot draw. So we have a blood vessel here. And inside the blood vessel you have blood. Blood is made up of cells. Just going to draw a red blood cell here. And plasma. So we have a cell in its environment. Now I'm going to go ahead and put some solute particles in here. So we're going to have, here's our solute particles in our plasma. And here's our solute particles in our red blood cell. What you notice is we have the same number of solute particles in the red blood cell as we have in the plasma. So we can say the red blood cell is isotonic to its environment, the plasma. This means no net water movement. Now that's not to say that water doesn't move. Water does move. So when I say no net water, what I mean is for every one water molecule that leaves the red blood cell, one water molecule will enter the red blood cell. So it's at equilibrium. We have no net movement of water. So it's a one-to-one situation here. This is normal. This is what we want. This is what your body is supposed to do. Okay? Now, let's take a look at what happens when things don't do what they're supposed to do. So let's look at a situation where we have a cell in a hypertonic environment. So I'm going to go ahead and draw our blood vessel again. Again, pardon my drawing here. And in our blood vessel, we have blood, which is made of cells and plasma. The plasma is the environment that the cell is in. Now, the red blood cell is going to have the same number of solute particles. That's not going to change. So if it had 3 initially, I'm just going to go ahead and keep with that same 3. So here's our solute inside of our red blood cell. Now, it's in a hypertonic environment. So if we're going to have a cell in a hypertonic plasma environment, that means we're going to have a lot of solute particles, right? Hypertonic. So our red blood cell is hypotonic, and our plasma will be hypertonic in comparison, okay? Now, which way is water going to go? We know that the solute particles can't move. So what's going to happen with the water? Water is always going to go from an area that is hypotonic to an area that is hypertonic. I'll give you a little hint. Water follows the salt. So I know our solute particle isn't necessarily salt. That doesn't matter, but if that's going to help you remember that, if you remember nothing else from this lesson, remember water follows the salt. So your red blood cell is going to leak water into the environment to try to reach an equilibrium. So your red blood cell is going to lose water. What's going to happen to our red blood cell? Red blood cell will lose water, causing the red blood cell to shrink. Right? It's going to get smaller. It just lost a bunch of water. So this is called crenate. And you're going to have a red blood cell. Again, I can't draw very well, but it's going to be, instead of this red blood cell that's going to be nice and round because it's full of water, because it's lost water, it's going to have this jagged appearance, and it's going to be smaller. It's going to shrink a little bit. So this is what a crenated red blood cell, pardon my drawing, is going to look like. Again, water is going to follow the salt. It's going to go to a more hypertonic environment. Okay, so now let's take a look at, well, I'll go ahead and tell you when this happens in real life. So in real life, when you have a situation like this where you have a red blood cell in a hypertonic environment, this would be when a person is dehydrated. Okay, they don't have a lot of water in the blood, so it's going to have a higher solute concentration because there's just not enough water in there anymore because you're dehydrated. So when a person is dehydrated, you can actually look at the microscope slide of blood and see these crenated red blood cells and probably the reason why is because that person is dehydrated. And the more dehydrated they are, the more created red blood cells you're going to see. Okay, so now let's take a look at a situation where we have a hypotonic environment. So we're going to draw our blood vessel again. And in the blood vessel, you have blood that has cells and plasma. Plasma is the environment. Now I'm going to stick with my solute particles, the same number. The solute particles inside the cells aren't going to change. It's what's in the environment that's going to be different from person to person and situation to situation. Okay, so we're going to put our cell in a hypotonic environment. So that means it's going to have less solute than the cell. So there's our solute particle. And that's going to say, okay, our red blood cell has more solute now. So this is now the hypertonic situation. And the plasma or the environment is the hypotonic situation. So which way is water going to go? Water is going to go from a hypotonic environment to a hypertonic environment. Water is going to follow the salt. So water, to try to reach an equilibrium situation, water will enter the red blood cell. So when a red blood cell or any cell is in a hypotonic environment, water will enter the red blood cell, causing it to swell. Obviously, you're putting fluid into an already full area, so it will cause it to swell and possibly burst. That's called lyse. So make sure you know what lyse and crenate means. I'm not going to draw a picture of a busted red blood cell because you probably know what a busted red blood cell looks like. It doesn't look like a cell anymore. So when would this situation happen in real life? This could happen when a person drinks too much water too fast in a very short period of time. You're basically overhydrating the blood, and the body can't offload the water faster than you're putting it in. And it could create a situation in the blood where red blood cells are now in a hypotonic environment and cause them to burst. What's the downside to this? Well, let's take a look and see what the actual role of a red blood cell is. Red blood cells carry oxygen throughout the body. If you just blew them all up because you overhydrated, you're no longer getting oxygen to the body, which means the body's going to die. So this could result in death. And it does. It has. So anyway, that's the situation that could happen in a red blood cell that is in a hypotonic environment. Doing too many things at one time here. Okay. So we've talked about this. We've talked about isotonic. We've talked about osmosis. There is a video on this, so I suggest you all click this link down here on your PowerPoint and take a look at that video so you can visually see what's happening there. Now, we talked about the fact that water is going to be able to stick to solute particles, and that makes them bound. But we never said why water has the ability to stick to those solute particles. Why does water stick in the first place? Well, it has to do with polarity. And if you guys remember back from the previous chapter, water is what's called a polar molecule, which means it's got a partial negative side, which happens to be where the oxygen is, and a partial positive, which is where the hydrogens are. And what that means is that polarity is due to the fact that these electrons right here are not being shared equally. The electrons are being, oxygen is pulling the electrons closer to itself, creating, and because electrons have a negative charge, that's going to create oxygen in this water molecule to have this partial negative charge where the hydrogen is a little electron deficient, and it's going to create a partial positive charge on the hydrogen. Now, these partial positives and negative charges are electrostatically attracted to ions in the in the plasma. So you can see your water molecule here. Remember, it kind of looks like Mickey Mouse here. You've got your partial positives on the hydrogen. You've got your partial negative on the oxygen. I'm not going to ask you what side anything's attracted to. I'm not worried about that. But if you have like salt in the blood, in the plasma, that salt is going to dissociate. It's an ionic compound, so it's going to break apart into sodium ions and chlorine ions, which are going to be charged particles. And these charged particles can be electrostatically attracted to one side of the water molecule. The chlorine, for instance, is electrostatically attracted to the other side of the water molecule. You can see the hydrogens are facing inward. Again, I'm not asking you any of that stuff. Bottom line, what I need you to know is that the water molecules are electrostatically attracted to other charged particles because water's polar, and that binds up the water, preventing it from being able to cross that barrier, that plasma membrane. Now, to speak on overhydration just a little bit, if you ever go to the hospital or see somebody who's got an IV bag, one of the things that you notice is it's not pure water in that IV bag. It is a saline solution. So, for example, a 0.9% sodium chloride solution. And if salt is so bad for a person, why are they giving you salt water in an IV bag? And that's because they're trying to keep it isotonic to the plasma. They don't want to create a situation where the plasma is going to be hyper or hypotonic to the cells. They want it to be isotonic to the cells. So that's why you have not just pure water in those IV bags. Now, overhydration occurs when a body takes in more water than it loses. To exceed the body's ability to excrete water, a young adult would need to drink in excess of 6 gallons of water per day. Now, in a short period of time, 3 to 4 liters of water, 2 quarts per hour in 20 minutes, or 4 bottles in 20 minutes. So again, that's also too much water. You've got to give your body time to offload that. There's a video again on osmosis, so I encourage y'all to take a look at that. This just goes through and shows you basically what we just drew. You've got your hypotonic environment, and you have your cell right here, which kind of looks like an orange with the green solute particles in there. So you can kind of follow that and make sure you understand what's going on there. This right here is showing you the net water movement. So you can see in an isotonic situation, there is no net water movement. Here you can see water is going into the cell from a hypotonic environment, and water is leaving the cell when it's placed in a hypertonic environment. Again, just pictures here. So one of your essay questions is describe the difference between hypertonic, hypotonic, and isotonic. So I'm telling y'all right now that if you were to write this phrase down for the description of hypertonic, you would get full credit. Okay, so when you're writing your essay question down, don't just put a solution that has more, or don't put more solute. You need to explain a little bit better than that what's going on. This is what, in a real blood smear, this is what lysed red blood cells look like. So you don't really see any red blood cells there. And this is what red blood cells look like that are crenated. The arrows are pointing to the crenated red blood cells. And this is what an actual blood smear looks like. You have got the white blood cells, which are represented by these large purple cells right here. You've got your red blood cells, which kind of look like little pink donuts. And then you have your platelets, which are these little bitty purple dots right here. So those are the cells that are found in blood. And then we will come back and take a look at active transport in the next video.